Hi everyone, welcome to Neuroscience for Non-Majors. This is week 3, unit 2. If you look on Canvas, I've posted the review from office hours, so hopefully that will answer your areas of confusion that you filled out in the exit survey during your first class. I've also posted a bonus lecture that you can watch if you want to that talks a bit more about the math behind voltage-activated ion channels as well as current flow across a neuron's membrane. Remember also to post your science fiction stories to Canvas by the beginning of class on Monday. Okay, let's get started on this week's material. This is the second week of overview describing kind of fundamental neuroscience topics, and in next week's lecture, we will finish these fundamentals, and after that, our lectures will start focusing on various different areas of like current neuroscience research and applications to different fields. But also all of these topics will be brought up like repeatedly throughout the course as we go through all the other material too, so yeah. Okay, let me share my screen. Okay, it's at the so this week's lecture topics will cover NMDA receptor structure and function, the strengthening of the synapse during long-term potentiation, and metabotropic receptors, and a different kind of receptor called a GABA receptor as well, which is also an ionotropic receptor, but we'll discuss that when we get to it. So in the first lecture, we discussed a type of ionotropic receptor that you can find on the dendrites of a neuron called the AMPA-type glutamate receptor, which opens when the neurotransmitter glutamate binds to it and lets positively charged sodium ions flow into the cell. Well, also found on these dendrites is another kind of ionotropic receptor that also binds to glutamate, and it's called the NMDA receptor. So the NMDA receptor is special because it acts as a coincidence detector in the synapse. It's only able to let ions flow through it for the short period of time when both the presynaptic and the postsynaptic cells are active. The reason it is only active when both the presynaptic and postsynaptic neurons at a synapse are active is because of two features. I'll start with the first feature. When glutamate binds to an NMDA receptor, the receptor opens just like an AMPA-type glutamate receptor. And if it opens, it would become permeable to sodium, potassium, but most importantly, calcium ions. But it has an additional feature that prevents these ions from flowing through. And the second feature is a magnesium ion plug. And it kind of sits, it's so big that it kind of sits in the channel of the NMDA receptor, and it's so big that it blocks the NMDA channel regardless of whether the channel is open or closed, and this prevents other ions from passing through. The magnesium ion is really, really attracted to the negative charge inside the cell, and it is only repelled when the cell becomes depolarized and more positively charged, like during an action potential. So it is only when, through the AMPA-type glutamate receptors, sodium ions flow into the postsynaptic cell and depolarize it, it's only then that the magnesium ion in the NMDA receptor is repelled by the positive charge inside and actually unblocks the NMDA receptor so that calcium ions can then flow in. So the only time that the NMDA receptor is able to allow calcium ions to flow through it is when the presynaptic neuron is releasing glutamate to open it and the postsynaptic neuron is depolarized like just before or during an action potential. This is why the NMDA receptor is considered a coincidence detector. It detects simultaneous activity in the presynaptic and postsynaptic cells. Now, why might this coincidence detector be useful? What could the purpose be of having calcium ions flow into the cell during this simultaneous activity? Well, The calcium ions are actually crucial for the process of adding even more AMPA-type glutamate receptors to the dendrites. And if there are more AMPA-type glutamate receptors on the dendrites of a neuron, the likelihood that the neuron the likelihood of the neuron becoming depolarized and firing an action potential with presynaptic release increases. So this NMDA receptor activity can actually strengthen the synapse by adding more AMPA-type glutamate receptors. And the mechanism of how it does this is when calcium ions flow through an NMDA receptor and enter a cell, the ions bind to an enzyme called calmodulin-dependent kinase 2, but you just need to think of it as CAMK2. You don't really need to know the name. CAMK2. The calcium ions bind to CAMK2. And this enzyme changes shape when calcium binds to it, and it grabs ATP, which is an energy-carrying molecule, from the cytoplasm inside the cell, and then it removes a chunk of the ATP containing phosphate and sticks that phosphate chunk onto a vesicle inside the cytoplasm that has an AMPA-type glutamate receptor on it, like in the vesicle. So it's phosphorylating it. The vesicle is now able to move to the membrane of the postsynaptic cell and fuse with it, so that the AMPA-type glutamate receptor is actually now on the exterior of the postsynaptic cell, and it can let more sodium ions flow through it if glutamate binds to it. If there are high-frequency inputs to the postsynaptic cell that are able to depolarize the postsynaptic cell, then the NMDA receptors are more active, and that allows the postsynaptic cell's response to individual presynaptic stimulations to become even stronger because more AMPA-type glutamate receptors will be added. This strengthening of a synapse with repeated activity at a high frequency is called long-term potentiation. And long-term potentiation is incredibly important for keeping the synapses that are important and frequently active strong. Now, glutamate is not the only neurotransmitter in the brain, and ionotropic receptors are not the only kind of neurotransmitter receptors in the brain. I want to briefly talk about GABA, which is another neurotransmitter, and then I want to talk about another type of receptor called a metabotropic receptor. But here we're talking about a GABA receptor. So if a neurotransmitter called GABA binds to ionotropic GABA receptors on the dendrites of a neuron, then those receptors open and allow negatively charged chloride ions into the cell. And since these chloride ions are negatively charged, they have the potential to cancel out other depolarizations. Like if sodium ions are flowing in or something like that, then they'll cancel that out, and then they'll inhibit the cell from firing an action potential. Okay, this is the final concept. A metabotropic receptor, which is also called a G protein coupled receptor, works a little bit differently from ionotropic receptors. Instead of being a channel protein, they are proteins which, when neurotransmitter binds to them, they allow the cell to become either more excitable or less excitable. They don't directly open, they aren't ion channels themselves, so they don't like directly open and let ions flow through. What they actually do is that when neurotransmitter binds to them, they have these little miniature proteins attached called G proteins. And when neurotransmitter binds, they release these G proteins. And then these G proteins go and do all sorts of different mechanisms, perform all sorts of different like functions and mechanisms in the cell. One of which includes like binding to other ion channels and opening those so that ions can flow through. But the metabotropic receptors themselves don't have ion channels in them. They indirectly open other channels or indirectly perform other mechanisms that can increase or decrease the cell's excitability. Ionotropic receptors, on the other hand, are actually ion channels, and when glutamate or any other neurotransmitter binds to them, they actually open and allow ions to flow through directly. So that's the difference between metabotropic and ionotropic receptors. And I'll go over all of this again in class. This is a shorter lecture, but this was a lot more, these are slightly more complicated mechanisms than the previous unit's material. So I'll go over all of this again in class tomorrow. And for this week's homework assignment, I want you to draw a comic, and it can be as long or as short as you want it to be, but it needs to have characters, whether those characters are humans or cells or neurotransmitters or receptors or like the world or whatever. And it needs to have dialogue. And also, but most importantly, it needs to demonstrate your understanding of at least 2 of the following topics: NMDA receptors and long-term potentiation, GABA receptors, and metabotropic receptors. And that's all for this lecture, and I'll see you soon. Okay, goodbye.